Welcome to Bio Bits and Bytes. Today we're plunging into the incredibly intricate world of RNA. These molecules are just so fundamental to life, and, well, we've got some truly exciting research that's showing how cutting-edge computational methods are not just helping us understand RNA, but are also giving us the power to design it in entirely new ways.
That's absolutely right. It's really a revolution happening at the molecular level with, pretty profound implications.
Okay, so in this deep dive, we're going to try and unpack two major advancements. First up, we'll be exploring ATOM-1, which is a brand new foundation model. It's making waves by predicting RNA structure and function using something called chemical mapping data.
Mm-hmm.
And second, we're going to delve into how deep learning is enabling us to predict the function of short, non-coding RNAs directly from their sequence, which, and this is pretty surprising, actually challenges some long-held scientific beliefs.
Both of these developments are, really game changers in how we approach RNA biology.
Absolutely. And to guide us through this fascinating landscape, we have an expert with us today. They're going to help us understand the science and, the real-world implications of all of this. And for everyone tuning in, we'll definitely be defining any key scientific terms as we go, so you can follow along on this exciting journey.
Sounds good.
So let's dive right in. This ATOM-1 model sounds like a pretty pivotal development. Could you maybe start by telling us what exactly is a foundation model in this context?
Yeah.
And what is this chemical mapping data uses?
Certainly. So in computational biology, a foundation model is basically a large, highly versatile model that's been trained on just a massive amount of data. The key idea really is that it learns very general, sort of fundamental representations of the underlying biological principles, in our case, those related to RNA.
Okay.
And because it's learned these broad principles, it can then be adapted, or as we often say, probed, for a whole wide variety of more specific tasks. You could almost think of it like a broadly educated expert who can then apply their knowledge to many different specialized areas.
Okay, so it's like a pre-trained intelligence for RNA,
Yeah.
sort of, that we can then ask specific questions to.
Precisely. Yeah, that's a good analogy. Now, chemical mapping data, that comes from experimental techniques that give us information about the local chemical environment and, importantly, the structure of an RNA molecule.
Right.
And imagine taking a series of very precise measurements right across the RNA, telling us how accessible different parts of it are to chemical interactions. These methods use chemical reagents that react differently depending on whether a nucleotide, one of the building blocks of RNA is paired up with another, or if it's unpaired, or maybe involved in some other kind of interaction within the RNA's 3D structure.
Ah, okay.
So the data we get essentially paints a picture, a kind of snapshot of the RNA's structural features when it's in solution.
That makes sense. So ATOM-1 is built using this detailed chemical snapshot of RNA molecules. What kind of things can it actually predict or, tell us?
Well, the researchers demonstrated some truly remarkable capabilities. One key achievement was its ability to predict the secondary structure of RNA with, frankly, impressive accuracy.
Secondary structure meaning how it folds back on itself.
Exactly, the base pairing patterns. They trained a very simple probe. Think of it as a small add-on module with only 257 parameters, which is tiny in machine learning terms. They trained it on the known secondary structure of just one RNA molecule. Its PDB identifier is 6WJR.
Okay, just one.
Just one for this specific probe training. Then they tested this probe on a completely different RNA called 8DP3 to see if it could predict how its bases would pair up.
And did it work? Was it successful?
Absolutely. If you were looking at figure 2 from their research, which shows the predicted base pairing probability right next to the experimentally determined structure for ADP3, you'd see a very, very clear match. And this is significant because it indicates that ATOM-1 has learned some really fundamental rules about how RNA sequences fold into these secondary structures. And importantly, it's learned these rules from that chemical mapping data primarily, with just a little bit of specific structural training for the probe.
Right.
What's exciting is that these learned rules seem to be broadly applicable to other RNA molecules. It suggests that ATOM-1 can give us really accessible and accurate representations of secondary structure.
So for researchers who might not have, say, huge computational resources or these massive sequence databases, ADAM1 could really democratize RNA structural biology, couldn't it?
Exactly. That's a great way to put it. It lowers the barrier to entry quite significantly for studying RNA structure.
Wow, that's incredibly efficient.
Hmm.
So, okay, what about the more complex three-dimensional shapes that RNA molecules can adopt, the sort of final folded structure?
Ah, yes, the 3D structure. This is where it gets really, really interesting, I think. The researchers also put ATOM-1 to the test for predicting the full 3D structure of RNA. They used a simplified or shallow version of a component called evoformer, and importantly, this version didn't rely on MSAs.
Okay, hold on. MSA free. Let's break that down.
Yeah,
What exactly are MSAs, and why is it such a big deal to not need them?
right, good question. So MSA stands for multiple sequence alignment. It's a standard technique in bioinformatics where you take the RNA sequence you're interested in and you compare it to a large collection of related RNA sequences from, other organisms, other species.
Okay.
By looking at the patterns, which parts of the sequence are conserved across many different RNAs, which parts vary, you can infer information about its structure and probably its function too. It relies on that evolutionary information.
Makes sense.
However, generating high-quality MSAs can be very time-consuming and computationally pretty intensive. Plus, for many RNAs, maybe ones that are newly discovered or don't have many close relatives known yet, it can be difficult or sometimes even impossible to create a reliable MSA. So yeah, it's often a bottleneck in the process of trying to understand a new RNA.
Right, so not needing an MSA could really speed things up and potentially allow us to study a wider range of RNAs, maybe ones we couldn't look at easily before.
Precisely. That's the key advantage. And what's so fascinating here is that even though the ADAM1 probe they used was much smaller and simpler, I think it had around 15 million parameters and just 2 layers, compared to methods like rowfold and rosettafold 2NA, which have tens of millions of parameters and many more layers.
Yeah.
It actually achieved higher global accuracy in predicting the 3D structure. They measured this using something called RMSD, which stands for root mean squared deviation. Basically, RMSD is a measure of how much the predicted structure deviates, on average, from the experimentally determined correct structure. Lower is better.
So a much smaller, simpler method, without needing these often difficult-to-get MSAs, performed better overall in predicting the 3D shape. That is quite surprising.
It is, yeah. And the story gets even more compelling. When they looked at the local accuracy, so how well the model predicted the structure of specific little regions within the RNA using a metric called LDDT, or local distance difference test.
Okay.
The ATOM-1 probe consistently outperformed a baseline network. This baseline had the same architecture but wasn't using the underlying knowledge, the embeddings, captured by ATOM-1.
Ah, so ADAM1 itself was providing the key information.
Exactly. It really highlights that ATOM-1 is providing these readily accessible and accurate 3D structural representations, and crucially, without the limitations of needing those MSAs. So RMSD tells us about the overall shape, the big picture, and LDDT tells us if the finer details in specific spots are correct. ATOM-1 seemed to do well on both.
This sounds like a really significant leap forward for researchers, trying to understand the intricate 3D architecture of RNA molecules.
It absolutely is. I think it makes high-quality structural predictions far more accessible to a much wider range of researchers, potentially accelerating discovery.
Okay, so we've seen how ADAM1 can predict both the secondary, sort of flat structure, and the tertiary 3D structure of RNA.
Yeah.
What about its function? Can it tell us anything about what an RNA molecule actually does in the cell?
Yes, indeed. And that's often the ultimate goal, right? Understanding function. The researchers also tackled the important challenge of predicting RNA degradation and reactivity, basically, how stable an RNA is and how it interacts chemically, which are crucial aspects of its function.
Right.
They trained another relatively simple probe network, this one with about 10 million parameters, using the learned representations, the embeddings we mentioned, generated by ATOM-1. And the results they achieved were, well, quite remarkable.
What did they find? How did it do?
This relatively small network, powered by ATOM-1, outperformed all 1,636 submissions in a major international challenge that was focused specifically on predicting RNA degradation and reactivity.
All of them. Wow.
Yeah, all of them. Figure 6A in their paper clearly illustrates this outperformance. And to really emphasize the crucial role of ATOM-1 itself, they also trained that baseline network, again, same architecture as their best probe, but without access to ATOM-1's underlying representations.
On the baseline.
The accuracy of this baseline was significantly lower. Its test loss was, I think, 37% higher, which is a big difference. It clearly indicates that it's the knowledge embedded within ATOM-1 that's driving this high prediction accuracy, not just the specific design of the probe network itself. So yeah, ADAM1 is really capturing something fundamental about how RNA behaves chemically.
That's compelling evidence.
Exactly. And it also appears to be quite robust. The researchers observed that the ADAM1 probe generalized very well to longer RNA sequences in the test set sequences that were about 30% longer than the RNAs it was initially trained on. Figure 6b, which compares the losses on the validation and test sets, supports this.
Okay, so it's not just good on the data it saw, it can handle slightly different longer molecules too.
That's right. It suggests that models built using ADAM1's representations are likely to be reliable even when dealing with RNA molecules that are somewhat different from those they've seen before during training.
So for our listener, the learner,
Mm.
maybe someone just getting into this field or trying to apply these tools, what's the key takeaway from all this about ADAM1?
Yeah, for the learner, I'd say ADAM1 offers a really powerful and importantly accessible shortcut to understanding the structure and potential function of RNA molecules. It can provide surprisingly accurate predictions, often without needing those complex and time-consuming multiple sequence alignments.
Right.
This makes exploring the world of RNA research more feasible and potentially much faster for a wider range of scientists. So you could imagine trying to understand how a complicated piece of origami the RNA folds up. ADA1 gives us a much faster and easier way to predict that fold, even if we've never seen that specific origami pattern before.
That's a fantastic analogy. Okay, great. Let's switch gears now and delve into our second main topic.
Yeah.
The, well, the surprising ability of deep learning to predict RNA function directly from its sequence.
Yes, this is a truly fascinating area that's prompting us to maybe reconsider some of our fundamental assumptions about how RNA works. A recent study explored using deep learning convolutional neural networks, or CNNs, to predict the function of short, non-coding RNAs based solely on their raw sequence, just the order of the A, U, G, and C nucleotides.
Just the sequence. That feels counterintuitive. I mean, I always thought RNA's function was primarily determined by its three-dimensional structure, how it folds up.
You're absolutely right to think that. The prevailing scientific view, the established understanding, has absolutely been that the secondary structure, how the RNA folds back on itself through base pairing, is a primary driver of its function. However, this research suggests that we can actually predict RNA function with a pretty good degree of accuracy using just a computational representation of the sequence itself without explicitly calculating those structural features first.
Wow.
This really makes us reconsider how much information is intrinsically encoded in just the linear sequence itself. It opens up possibilities for functional annotation even when we don't have detailed structural information, which is often the case.
That's a pretty significant challenge to the traditional view then. How did they go about investigating this? What was the setup?
Well, the first critical step, as always in machine learning, was to create a comprehensive and high-quality data set. The researchers started with a large collection, over 650,000 RNA sequences from the RFAM database, which is a well-curated resource that classifies RNAs into families based on conserved sequences instructions.
It's a standard resource.
Exactly. They then applied some careful filtering to focus specifically on short non-coding RNAs. They excluded longer RNAs and also classes where function was clearly dependent on the RNA's length to avoid confounding factors. This resulted in a novel data set of over 306,000 sequences across 88 different RFAM families. So a good-sized, focused data set.
Right, so really substantial data set of
Yeah.
these short regulatory RNAs. What do they do with all that data then? How did the deep learning come in?
They then employed a fairly standard deep learning CNN architecture. You can think of these networks as being sort of inspired by how the visual cortex in our brain processes images. They consist of multiple layers of convolutions. These are mathematical operations that help the network learn specific patterns within the input data, which in this case is the RNA sequence represented numerically. These convolutional layers are typically followed by things like ReLU activation functions, max pooling layers, which help to kind of summarize information, reduce complexity, and dropout layers, which are a technique to prevent the network from basically just memorizing the training data, helps it generalize better.
Right, prevents overfitting.
Exactly. They used both 1D convolutions, which are well suited for processing sequential data, like k-mers, and also 2D convolutions for another way of representing the sequence called space-filling curves, which we can touch on. Think of these layers as the network's way of learning increasingly complex patterns in the sequence data, starting with simple features and building up to more intricate relationships.
Okay, let's pause there for a moment. You mentioned K-mers. Can you explain what those are for us?
Oh, sure, yeah. A K-mer is simply a contiguous subsequence of K nucleotides within the RNA sequence. So, for example, if you have the RNA sequence AUGC and you're looking at tumor, so K equals 2, the K-mers would be AU, UG, and GC.
Got it. Short snippets.
Exactly. Using K-mers allows the network to learn about short, local patterns and motifs within the sequence, which can often be functionally important.
Okay. And you also mentioned space filling curves. That sounds like a more abstract way to represent the data.
Yes, it's a bit more abstract. Space-filling curves are a mathematical technique, essentially a way to map a one-dimensional sequence, like our linear RNA sequence, into a two-dimensional space, like an image.
Huh. Why do that?
Well, the idea here was to explore if this 2D representation could implicitly capture some information about the RNA's potential structure. The thinking was that by placing nucleotides that might be far apart in the linear sequence but actually close together in the folded 3D structure, closer to each other in this 2D representation, the 2D CNN might pick up on structural relationships.
Interesting approach. Sort of trying to sneak structure in without explicitly calculating it.
Kind of, yeah. It was an exploratory approach. And they also had to address the practical issue that RNA sequences come in different lengths, right? So to handle this for the CNN input, they explored different padding strategies, basically adding extra symbols to the ends of shorter sequences to make them all the same length for the network. They experimented with filling these extra spaces with random nucleotides, a constant symbol, or even a new, unique symbol just for padding.
Okay, so they tried quite a few different ways to represent the sequence information for the deep learning model. What were the most important results then? What really stood out from this research?
Several key findings emerged, I think. First and foremost, they discovered that using those K-mer encodings actually performed very well in predicting the function meaning, classifying the sequences into their correct RFEM families. They achieved high accuracy just based on those short sequence patterns.
Okay, so K-mers worked well.
They did. Second, and this was quite surprising, they found that these K-mer encodings were significantly more robust to the addition of what they called boundary noise.
Boundary noise.
Yeah, this noise was meant to simulate the uncertainty that can occur in real-world sequencing experiments, where you might not be absolutely certain about the exact start and end points of an RNA sequence you've sequenced.
Ah, okay, practical problem.
Exactly. And figure 8 in the paper shows that as they added increasing amounts of random nucleotides to the beginning and end of the sequences, the performance of the camera-based methods only declined slightly. Whereas methods that rely on predicting secondary structure information first, like other tools called Eden and NRC, they experienced a much more substantial drop in accuracy.
That's a really interesting point about robustness to noisy data. It suggests that in situations where you might have less than perfect sequencing data, maybe looking directly at short sequence patterns is actually more reliable than trying to infer a precise secondary structure first.
That's certainly a strong implication, yeah. Third, kind of related to your earlier point, they found that using those space-filling curves as a proxy for secondary structure actually resulted in weaker performance compared to the K-mer encodings.
Oh, really? So the attempt to implicitly capture structure didn't pan out as well?
Not in this case, no. Which, again, really challenges the idea that explicitly modeling RNA structure is absolutely necessary for predicting function, at least in this specific context of short, non-coding RNAs. It suggests the sequence itself might hold more direct functional information than we previously thought.
So the sequence itself might hold more functional information than we previously thought, at least for these short RNAs.
That seems to be a key implication of their findings, yes. And finally, they also investigated using a technique called Monte Carlo dropout. This is a way to estimate the uncertainty of the model's predictions.
Why is uncertainty important?
Well, it allows the model to sort of say, I'm not sure about certain predictions. This let them identify and potentially reject classifications that the model was less confident about, maybe sequences that weren't actually functional RNAs or ones where the prediction was just ambiguous. Figure 9 demonstrates their ability to recognize potentially non-functional RNA using this approach. And Table 4 and Figure 10 showed that by filtering out these uncertain predictions, they could actually improve the overall accuracy of their functional predictions on the remaining, more confident classifications. So it adds a layer of reliability.
So for the learner, again, what's the big picture significance of this research on deep learning and short non-coding RNAs? What should they take away?
Yeah, for the learner, I think this research offers a potentially much faster and computationally less demanding way to gain insights into the function of short, non-coding RNAs, especially perhaps in situations where you might have noisy experimental data or where determining the RNA structure is technically challenging or time-consuming.
Right.
And it also presents a new way of thinking about RNA function, really highlighting that the linear sequence itself contains a wealth of information about what an RNA molecule does, possibly more than we've traditionally recognized, at least for these types of RNA.
That's truly groundbreaking stuff, potentially shifting how we approach annotation. Now, we also briefly mentioned earlier the idea of not just understanding existing RNA, but actually designing new RNA molecules.
Oh.
How are these computational methods contributing to that? That sounds even more futuristic.
Absolutely. And it's happening now. One particularly exciting area here is the application of generative adversarial networks, or GANs, in the field of RNA design.
Okay, GANs. We've heard about them being used for creating like realistic images and
Yeah.
videos, right? How do they work for RNA sequences?
The core principle is actually quite similar. A GAN typically consists of two neural networks that are trained together in an adversarial sort of competitive way. You have a generator network that tries to create realistic-looking RNA sequences.
Okay.
And you have a discriminator network that tries to distinguish between real known RNA sequences and those fake ones generated by the generator. Through this ongoing competition back and forth, the generator learns to produce RNA sequences that become increasingly similar to naturally occurring functional RNAs or RNAs with desired properties.
So it learns by trying to fool the other network.
Exactly. For example, a specific model called GARDN, that's GARDN, for Generative Adversarial RNA Design Network, has been developed specifically for RNA. It can learn complex representations capturing aspects of both RNA sequence and predicted structure, and then use this understanding to generate entirely novel sequences that are predicted to possess specific functional properties.
Like what kind of properties?
Well, the researchers behind Garden actually demonstrated its ability to generate things called toehold switches. These are clever little RNA devices used in synthetic biology with improved performance characteristics, like better switching between on and off states.
Ah, okay, so designing molecular switches.
Precisely. And they also highlighted the use of another predictive model called Sandstorm, kind of working together with Garden. Sandstorm acts as a sort of quality control or predictor, allowing researchers to further optimize the RNA sequences generated by Garden to achieve, say, desired on-off ratios in these toehold switches. How well they turn gene expression on and off in response to a specific trigger RNA.
So it's a combination. One network generates ideas, the other predicts how well they'll work.
Yeah, it's a powerful combination of generative and predictive models. It allows for the design of RNA molecules with potentially enhanced or even entirely new functionalities. And interestingly, just as an aside, there's an analogy to the protein world, where GANs have also been used successfully for tasks like predicting missing regions in protein structures, which suggests potential for similar applications in RNA structural biology, too.
That's a really powerful combination of tools. Generative models to explore the vast space of possible RNA sequences, and predictive models to guide that exploration towards molecules with the functions we actually want.
Exactly. And building on this idea of predicting function and even improving upon it, we also encountered the concept of RNA language models, or LMs.
RNA language models. Okay, that immediately makes me think of the language models used in natural language processing,
Yeah.
like GPT for understanding and generating human text. Is it similar?
The underlying concept is indeed very similar, yeah. These RNA language models are trained on massive data sets of RNA sequences, just like NLP models are trained on text to learn the grammar of RNA. The statistical patterns, the relationships between the different nucleotide bases, the sequences that tend to appear together.
Okay, learning the rules of the RNA language.
Exactly. One of the research sources we looked at mentioned a new and valuable resource called the GARNET database, which actually links RNA sequences to the optimal growth temperatures of the organisms they come from.
Oh, interesting. Why temperature?
Well, temperature puts a strong selective pressure on RNA structure and stability. So this database was used to train these RNA language models, effectively teaching them about RNA sequence patterns in the context of environmental adaptation, specifically thermostability.
Okay, learning from nature's solutions to heat problems.
Precisely. And one particularly interesting technical finding was that breaking down the RNA sequence into overlapping triplets of nucleotides, for example, if you have AUCG, you'd look at AUC, and UCG was found to be especially effective for training these models. more so than looking at single bases or pairs.
Triplets. Why triplets? What's the advantage of looking at the sequence in chunks of 3?
It's thought that these triplets might better capture the local sequence context that influences how RNA folds and how it interacts with other molecules like proteins. It's related to the codon structure in protein coding, but seems useful even for non-coding RNA. By learning the patterns of these 3 nucleotide units, the language model can develop a more sophisticated understanding of the underlying rules governing RNA sequence and function.
That's a fascinating insight into how these models learn. So how are these RNA language models being applied then? Can they actually improve RNA function?
Yes, that's one of the key applications being explored. Predicting specific mutations, small changes in the RNA sequence that could enhance a desired RNA function. A great example is thermostability, the ability of an RNA molecule to maintain its structure and function at higher temperatures.
Okay, making RNA more heat resistant.
Exactly. The researchers demonstrated this by first training a general RNA language model and then fine-tuning it specifically on families of RNA known to be stable at high temperatures, like the 23S ribosomal RNA, rRNA, found in heat-loving organisms, thermophile.
Specializing the model.
Right. Then they used this specialized fine-tuned model to predict mutations in the 23S rRNA of a common lab organism like E. coli, which isn't naturally heat-loving that might make its ribosomes more resistant to heat. They're essentially transferring knowledge learned from extremophiles to engineer a more common RNA.
How do they pinpoint which mutations might work?
It's a clever approach. They essentially compare how likely the language model thinks a particular RNA sequence is, both with and without a specific potential mutation. They look at the difference in these likelihoods, or log likelihoods technically, between the general model and the heat-specialized model. A big positive difference suggests the mutation is favored by the thermostable grammar the model learned.
Ah, okay, comparing probabilities from the generalist and the specialist model.
Exactly. Figure 6b in their paper shows how they calculated this relative log likelihood, and they even identified specific mutations in the E. coli 23S rRNA, like changing a uracil to a cytosine C at position 2477. And crucially, their experiments, shown in the paper, provided actual experimental evidence that this specific mutation did indeed increase the thermostability of the E. coli ribosomes.
Wow, so prediction followed by experimental validation. That's powerful. And how do these computationally predicted beneficial mutations compare to ones we might find just by, say, looking at natural sequence variations across different species? Are they the same?
That's a great question. And interestingly, the mutations predicted by these RNA language models often differed from those that would be identified by simply comparing the 23S RNA sequences of thermophilic and non-thermophilic organisms.
Oh,
Yeah, it really highlights the novel insights that these deep learning models can provide. They're not just recapitulating what we might figure out from straightforward evolutionary comparisons. They seem to be capturing more subtle sequence function relationships. And importantly, while some other powerful computational methods like graph neural networks or GNNs often require detailed information about the RNA's three-dimensional structure to work well,
right.
these language models demonstrate they can learn important functional features directly from the one-dimensional sequence itself.
It truly sounds like these computational tools, from ADAM1 to GANs to these language models, are opening up entirely new avenues for both understanding the fundamental biology of RNA and for engineering RNA molecules with novel and improved functions.
They absolutely are. It's a really exciting time. From gaining a much deeper, faster understanding of RNA's intricate structural and chemical properties to the really thrilling possibility of designing completely novel molecules with tailored functions, these computational approaches are proving to be incredibly powerful and genuinely transformative for the field.
This has been an incredibly insightful deep dive into the computational world of RNA. We've explored how groundbreaking models like ADAM1 are giving us really unprecedented access to understanding RNA structure and function, often bypassing previous bottlenecks. We've seen how deep learning is challenging our long-held assumptions about the relationship between sequence and function, particularly in short non-coding RNAs. And we've touched on the remarkable potential of GANs and RNA language models for actually designing and optimizing RNA molecules for specific purposes. It's clear, I think, that we're witnessing a real paradigm shift in how we study and potentially manipulate these essential molecules of life.
Indeed. The ability to rapidly gain crucial insights, accurately predict functionality sometimes just from sequence, and rationally design novel RNA molecules. It really opens up tremendous possibilities. You can think about developing new and more effective therapeutics, creating faster and more accurate diagnostics, and building innovative new tools for synthetic biology. The potential impact is huge.
We sincerely hope this exploration has sparked your curiosity and maybe provided you with a deeper appreciation for just how dynamic and incredibly important the field of RNA research has become, especially with these powerful computational advancements driving it forward. Thank you so much for joining us today and sharing your expertise.
My pleasure. It was great to discuss it.
And thank you for joining us on Bio Bits and Bytes.